Welcome student. I am Professor A.S. Rathod, Department of Genetics and Plant Breeding, Kekiwa College of Agriculture, Nasik, affiliated to Mahatma Phule Krishi Vidyapith, Rahuri. Today is our topic for lecture, that is the landmarks in plant breeding. Means in that the from the ancient up to the 2020, which are the different research and which are the different achievements made by the different scientists and the different plant breeders, we can see one by one. In broad sense, history of plant breeding can be divided into four parts. To the landmarks is also the history of that the plant breeding. In that the landmarks, the we can explain the different achievements of that the different scientists, and that point is came under the history of that the plant breeding. Thus we give the title of that our lecture, that is the landmarks in plant breeding. In that the pre Mendelian era means before the 19th of the century. Then Mendelian era 19 to the 1920. Then post Mendelian era 1921 to the 1950. Then modern era after the 1950. Pre-Mendelian era means 9000 before Christ. First evidence of plant domestication in the hill above the Tigris river. Then the 3000 before Christ, domestication of all important food crops in the old world. Then the 1000 before Christ, domestication of all important food crops in the new worlds. Then the 700 before Christ, Babylonians and Assyrian hand pollinated date palm artificially. Then in that day 1665, Robert Hooke, native of that England, first described this cell and known as father of cell biology. In that day 1694, Cameres of Germany first to demonstrate sex in plants and suggested crossing as a method to obtain new plants. Then Thomas Fairchild developed first interspecific hybrids between Sweet William and Carnation species, popularly known as a Fairchild's mule. Then 1727, the first plant breeding company was established in Germany by Wilmorins. Then in that day 1753, Carlos Linnaeus published his book Species Planetarium, including binomial nomenclature of that the plant taxonomy, means they give the name to that the species. Then 1766, Joseph Cultures from the Germany demonstrated that hybrid progeny traits from its both parents, and he produced scientific hybrid of tobacco. Then 1890, first used artificial hybridization in fruit crops. Then 1819, sheriffs utilized pure line selection to develop a new oat cultivar. Then John Lee Coulter developed the concept of progeny test and individual plant selection in a cereal. There in that the 1856, D. Vilmorin from French further elaborate the concept of progeny test and use in sugar beet. After that, Darwin published Origin of Species, their book. Then the most important eminent scientist of the genetics, that is the G.J. Mendel from Austria, published his discoveries in experiment in the plant hybridization and give the law of inheritance in Garden P. Then Rimpu firstly made inheritance cross between Retica mustum and cereal producing Reticle in that day 1890. Then in that day Mendel era, in that day 19 D. Varies, Correns and test marks rediscover Mendel's law of inheritance independently. Then Nilsson from Swedish introduced individual plant selection method in Sweden. In that year of 1903, Johnson proposed the pure line theory and also coined the term genotype, phenotype, and that the pure line. In that 1908, C.B. Davenport first proposed dominance hypothesis of heterosis. 1908 GHL EME's proposed over dominance hypothesis in maize. In next, Nielsen given the explanation of multiple factor hypothesis for grain color by using pedigree and the bulk breeding methods. Then scientists GHL first used the term heterosis for hybrid figure. Then scientist Jones in that the year of 1917 developed first hybrid maize and proposed dominance of link gene hypothesis. That the 1919 A. H.K. Garbers and Gave's initial ideas about recurrent selection. They first suggested use of synthetic varieties for commercial cultivation in match. Then post Mendelian era in that the year of 1923, Sykes proposal linkage between quantitative and qualitative traits. That in 1926 EME's and H. Mangelsdorff first discovered gametophytic incompatibility in Nicotina sanderi. In Vavilo identify 8 main centers and 3 subcenters of crop diversity. Scientist Mueller proposed mutation in flies using X-ray. Scientist Karpencheko developed first intergeneric hybrids between radish and cabbage in Russia. And that in 1928, scientist L.J. Stadler first used X-ray for induced mutation in barley. In that in 1933, Rhodes discovered cytoplasmic male sterility, that is the same as in the maize crop. Scientist Dustin discovered colchicines. Scientist Vavilo published the scientific basis of plant breeding. In that the 1935, Nagaham proposed the origin of tetraploid species of Brassica using triangle. Scientist C.H. Golden first suggested the use of single seed descent method for the crop improvement. The scientist C.M.T. Jenkins described the method of recurrent selection. Scientists Avery, MacLeod and McCarthy described the transforming principles and suggested that DNA is not a protein, is the hereditary material. In that in 1945, scientist F.H. Hull coined the term of recurrent selection and over dominance in that maize crop. And the last, that is the scientists Hughes and Backwalk first discovered saurophytic cell in compatibility in the Crepis foetida. So from the pre-Mendelian era, in the Mendelian era, after the post-Mendelian era, and up to the 1950s. So different scientists, they have to hard work in that to research in respect to that the plant breeding and to develop a different techniques and the different crop improvement aspects. So, thank you.